So we're going to continue on with proteins. This is the second video of the proteins chapter. So I mentioned earlier the disulfide bonds were able to stabilize the structure. This could be stabilization of tertiary structure or even sometimes quaternary structure. And and so the disulfide bonds are only able to be formed under oxidizing conditions. The reducing conditions of the cytosol are going to break disulfide bonds. And so we will never see disulfide bonds uh, in the cytosolic portion of a protein. Um, so you can see that the that the disulfide bond is just between two cysteines. Uh, so so you just take away those hydrogens and the sulfurs just interact with each other, and they're going to create a bond, a disulfide bond that that will stabilize the tertiary structure of the protein. So just to review, the four levels of protein structure are primary, secondary, tertiary, and quaternary. The primary structure is the left to right reading of the sequence of amino acids, and the primary structure is formed by peptide bonds between the amino end of one amino acid and the acid end of another amino acid. The secondary structure is going to be hydrogen bonds, and hydrogen bonds specifically within the backbone of the uh, the peptide chain, or the amino acid chain. Um, the polypeptide chain is sometimes what we call it. Okay, so um, tertiary structure could be hydrogen bonds. It could be charge-charge interactions. It could be van der Waals forces, but a lot of non-covalent bonds, and even some covalent bonds when we're talking about the disulfide bonds. Uh, so this is going to be between R groups and the backbone, R groups and R groups, backbone and backbone, and so the van der Waals forces are going to keep the hydrophobic amino acids on the inside of the protein, and the overall structure of the protein is going to be held together but able to be changed. And so things like pH or maybe phosphorylation will add a big negative group that will suddenly start a charge-charge interaction with some other place on the protein. And so the tertiary structure is a little bit more fluid than the primary and secondary structure. The tertiary structure can be changed by um, those non-competitive inhibitors, activators, cofactors, coenzymes, um, pH, temperature. There's a lot of things that can change the tertiary structure. And then remember that, and tertiary structure is the overall 3D structure or the globular structure of the protein. So then remember that quaternary structure is between more than one uh, gene product. So you've got two proteins, two or more proteins interacting with each other to create a complex, protein complex that allows it to do its function. The one you're going to remember here is hemoglobin is made up of two alpha hemoglobin subunits and two beta hemoglobin subunits. So these alpha globins and these beta globins are going to interact with each other to create an overall 3D structure that is the functional protein. And so again, these are going to be non-covalent or covalent bonds between subunits, and the covalent bonds are again going to be disulfide. Okay, what determines which amino acid is inserted? That's the DNA has a sequence, A, C's, T's, and G's, that those A, C's, T's, and G's are then transcribed into RNA, and then the RNA goes into a ribosome and gets read by the ribosome to uh, insert the correct amino acid depending on what the three letters of the triplet are. So in this case, like let's say you had a G, an A, and a G. GAG would lead to aspartic acid. But um, um, say like GC anything will lead to alanine. And there are going to be acidic um, amino acids, uh, aspartic acid and glutamic acid. There are going to be basic amino acids, arginine, histidine, lysine, and there are going to be polar and nonpolar. So the nonpolars are alanine, valine, methionine, isoleucine, proline, leucine, tryptophan, and phenylalanine. And you don't have to necessarily memorize all of these, but a a good um, mnemonic to remember the uh, the hydrophobic amino acids is family VW. Family VW. So if you're driving around in a microbus, you'll remember it. Okay, so how do we know what the correct structure of a protein is? Well, when it's doing its job, that must be correct. That's really all we can say. What is correct? How do we know that a correct structure even exists? Well, when it's doing its job, it is the correct structure. But how does the cell know that it's in the correct structure or not? Is are there any hydrophobic amino acids exposed to the cytosol or exposed to an aqueous environment? And if the answer to that is yes, then the cell is going to take action to refold that protein, maybe upregulate some heat shock proteins or maybe tag that protein for destruction um, because the consequences of not taking care of it is pretty bad. So, sickle cell anemia, for instance, is just one hydrophobic amino acid. It's a single base pair substitution that uh, exchanged a glutamic acid for a valine. So it was a charged amino acid, and now it's a non-polar amino acid. But because it was charged, it was on the outside of the protein, and so now you've got this hydrophobic amino acid on the outside of that protein. Well, the oil and water don't like to mix, and so now that hydrophobic amino acid it interacts with another hydrophobic amino acid on, say, another hemoglobin, and so now you've got this chain of hemoglobin, 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 and they create these insoluble aggregates that can literally change the shape of the cell because they've all glommed onto each other. Remember that example I gave, the albumin was supposed to be liquidy and clear, but when we denature it, we expose a whole bunch of hydrophobic amino acids. Um, we we straighten out the protein, And now there's all these hydrophobic amino acids that used to be on the inside of the protein, now on the outside of the protein. And so what do? They grab onto each other so that they can get away from any water. And that's how we create these non-soluble aggregates of the egg white of a fried egg or a boiled egg. And so that's how the cell knows whether the protein is folded correctly is are the hydrophobic amino acids on the inside of the protein. So the anafensin experiment is basically he broke all of the uh, all of the disulfide bonds of a protein. The protein was ribonuclease, and he broke all of the disulfide bonds by treating it with um, 8 molar urea and and beta mercaptoethanol. So beta mercaptoethanol was able to break all, so that's going to create a very reducing environment, and so it broke all of the disulfide bonds, and then the 8 molar urea um, broke all of the hydrogen bonds in the protein, and so uh, he was able to take away all of the secondary and tertiary structure, and then he reversed it, and he reversed it by adding um, an oxidizer and removing the urea. But what he found was that if he removed the urea first and then added the oxidizer, then he got a protein that was pretty much like it was supposed to be, original protein. But if he added the oxidizer first and then removed the urea, then he got a scrambled protein, a protein that was not able to perform the function of the ribonuclease. And so, what can we conclude from that? We can conclude from that that the whole purpose of the disulfide bonds is just to lock in that structure. It's the uh, sequence of the amino acids that determines the protein structure, and that the purpose of the disulfide bonds is just to lock in whatever they folded up into. Okay, so move on to video 3, and this concludes video 2.